Alrighty, so I think we are pretty much leveling off here. So I'm going to take this time to introduce our speaker today. Before I do so, if you are not at the Carey Institute and want to drop into the chat where you're joining us from, it's always great to see kind of where our audience is joining in from. And also just a reminder, if you have issues with kind of the tech side or you're not seeing the screen or anything, please let us know in the chat. But if you have questions for our speaker, please put that in the question and answer section, and I will help moderate those at the end of the talk. All right, so today it's my pleasure to have Dr. Kyle Wickings joining us today. Kyle is a professor at the Cornell University. He's in the Department of Entomology, and most of his work is up in the Geneva, New York campus, so he gets a chance to work a lot with kind of the community out there. Before joining Cornell, he got his PhD at the University of Georgia and also did some postdoc stints at the University of New Hampshire and Michigan State University. I am really impressed with the work that Kyle does because I think he has managed to kind of cross these areas that are sometimes difficult for scientists, where he's doing really great basic scientific research, but also really investing in working with shareholders and working with the public to translate that research into really solution-driven science. And I think that's really exemplified by the fact that, you know, his position is part split research as well as part extension at Cornell. And so I feel like I'm learning from Kyle about how to translate what I'm doing on the research side into working with the public and really trying to solve some of these bigger ecological problems. So I'm really excited today to have him talk to us about his research program, and I will let you take it away, Kyle.
Thanks, Jane. Thanks for the nice introduction. And is my audio okay? It was saying I had an unstable connection to match my unstable personality.
You sound great on my end.
Great. I'm going to share my screen. All right. Yeah, well, thank you very much for the introduction, Jane, and thanks for having me out here. I've taken over one of the offices here at Carey, so I am here. I've also learned how to use the espresso maker, so man, I'm just like settled right in. But I'm going to talk today about some work that my lab's been doing looking at the role of invertebrates in soil processes, and I'm really interested in how invertebrates affect microbial processes in soil. So we'll talk a lot about that. But just to give some background and some context to where I'm coming from, at Cornell, I have a research and extension appointment, as Jane said, and my extension appointment is with turfgrass managers. So it's a brand new commodity for me in terms of where I was trained, and I came out of sort of ag systems, but in agroecology, but turfgrass was really new for me. Trained as a soil biologist and soil animal ecologist, but I've always been interested in how they function in managed ecosystems. So my extension role at Cornell focuses on sustainable management of below ground pests. And that's where it connects with turfgrass, because turf has a lot of below ground insects with really cool ecology and biology, but also some very relevant management concerns that my job is to help that industry solve those. So as Jane kind of hinted at, our work kind of walks that line between basic and applied, and a lot of the grads and postdocs in my lab are the ones doing the really exciting ecology. And I'll talk a lot about their work today, as well as some of the stuff that I do that's on the more applied side of things and trying to bridge those worlds. So yeah, so that's kind of where I'm coming from. And we treat turf grasses as really cool, heavily managed below ground hotspots. So it's a really neat system, I think, to work in, despite all the management constraints that I'll touch on today throughout the talk. I kind of want to talk about some of these trade-offs between services and disservices in the ecosystem service framework of grasses. Like, what do they provide, and what are some of the constraints we have to deal with in those systems? It's going to have a very below ground focus, just because that's where I'm coming from. We'll look at some of our work on soil macroinvertebrates and how they influence microbial communities and soil processes in lawn ecosystems. We'll talk then about some of the work that we've been doing on management and how management can change those interactions and change below ground communities, particularly pest management. And I want to then talk about some of the work we've been doing looking at insect pests and how left unmanaged, how they can affect some of the soil processes that we're interested in preserving and optimizing in soils. There's some interesting feedbacks with pests that we didn't really expect to see. So I want to cover some of that with you all as well. And then I thought it would be fun to talk about some of my extension programming because, I'm kind of working in this world that wants really applied quick answers to solving pest problems, but also it's an opportunity for me to work with this stakeholder group on soil biology and soil health and sustainable management. So I thought it'd be helpful to talk about how I've been trying to bridge that in my program and what kind of programming we've been doing. So when we think about pest management and managing critters in soil or managing plant health, this is kind of the framework we typically think of. We have entire journals just focused on this pipeline, intervening to stop pest and pathogen populations and the damage they cause to plant systems. So you can spend your whole career just in this vein here, focusing on how to effectively manage pests. But the approach that we've been taking in our lab is to really think of this a bit more broadly and look at trade-offs. So there's growing interest in the turfgrass industry in sort of harnessing biological control, beneficial ecosystem services, and trying to optimize those. So we try to walk that line and ask questions about, well, how is the way we're managing pests also influencing some of the beneficial processes and biota that are in the system? So we'll keep coming back around to this framework, this sort of service and disservice balance that we try to seek in our work. So to talk about some of the benefits and those services, there's a lot of controversy around this, especially when you get into like lawn care and that component of the turfgrass industry. There's lots of data citing the benefits of turf grasses in filtering water, preventing soils from eroding, creating recreational space for athletics and just general outdoor activity. Huge cultural and educational value. I think turf grasses have some untapped, some already very present. They can be reservoirs for biodiversity, especially in heavily urban areas. And there's even data showing they can be important CO2 sinks under certain conditions. But it's interesting because when you look at the drawbacks and some of the disservices that some of these systems can provide, it almost is like a mirror image in many ways. So they can be water intensive systems, depending on how they're managed. They can be really important sources for pollution and nutrient runoff. They can support lower wildlife habitat. They're points of exposure for pesticides, depending again on how they're managed. Which can lead to biodiversity suppression, and also very much depending on how they're managed can be sources of CO2. So like walking this line between these pros and cons and trying to figure out how to optimize them has been a major challenge. And we've been thinking in my lab a lot about these last couple bullets: biodiversity and biogeochemistry and carbon cycling, carbon storage. Surprisingly, there's an uptick in interest around carbon storage in turf grasses. So we'll touch on a little bit of both of those today with some of our works. So in sort of thinking about these systems and their potential, you can really look at them the way you would look at a native grass system in general. So they can support really high below ground plant biomass. They're perennial systems. They're unlike ag systems in that they're not experiencing a lot of cultivation. They're cultivated, but not in a complete turnover kind of way, like a till versus no till kind of framework. But they can support really high root biomass, way much more biomass below ground than above ground, and can have potentially very high soil biodiversity. And I'm thinking really in the invertebrate world there, but I know that there's great data supporting that for microbial communities as well. So there's been a lot of interest in that potential, and a lot of people, whether it's from an urban ecology standpoint or really applied turf management standpoint, have tried to characterize, well, what is just the carbon storage potential of these ecosystems, comparing across cities, comparing with different turfgrass types, whether it's golf courses or residential settings. And there's some interesting takeaways that have come from this work. I mean, we know that turfgrass
Hi, everybody. Clearly having a little bit of a technical issue here, so sorry about that. Stick with us. I think hopefully we can get Kyle back on here. I think our internet is a little, just a little jumpy today. So stick with us and hopefully we'll be back in a moment.
Hey, everybody.
Hey, Kyle, sorry about that. I think a lot of people here at Cary had an internet cutout. So, but I think this is where we were leaving off. So,
Great. Yeah, I was gonna ask how much of my rambling people missed. So,
what's the slide right before this?
this one here.
This one. I think this is where you were just getting into how much of the cover is turf grass. So
Okay, great. So I didn't talk too much before it cut out.
no.
So yeah, we know that these residential turf grasses are a pretty large part of the footprint of terrestrial systems. So 40 million acres of turf grass in the United States. It's a really significant land use type. And if anybody knows anything about its management, it's like the Wild West in terms of how it's actually managed. But there's been a number of good studies done looking at its carbon storage potential, and some work even by Rattan Lal looking at this, showing that it can support quite a bit of carbon storage compared to other ecosystems. But there's so much variability behind that and in terms of how much and what drives its potential, especially what drives some of the potential carbon losses from the ecosystems. We know that it's really variable across the country as well, Northeast versus Southwest. There are some places that turf grasses just don't make a lot of sense. And there's a lot of unknowns around some of the management practices and how they might influence these cycles. So fossil fuel use in the system can be high, especially because we're using a lot of 2-cycle engines that are not the most fuel efficient equipment out there. Fertilization practices are all over the place in terms of what people do, especially in the residential area. Pesticide use, we've been looking at this, like how does pesticide use affect the biological processes involved in carbon cycling? Irrigation, organic matter management, this is huge in turf grasses. Some systems, all of the organic matter inputs that are being managed are exported out of the system for sort of like aesthetic and sanitation purposes. So a lot of the different factors of management components of turf grasses can drive variability in whether or not they can actually serve as a carbon sink. And there's even data showing that some of these processes and practices can turn them into a carbon source. So we've been really interested in this, and even just this one particular service, understanding what role do biota have in it and how does the way we manage it impact its potential. So in terms of how the processes are driven in these systems, we think a lot about invertebrates and other soil biota. And we've been asking the question, what role soil fauna play in organic matter cycling in turf grasses? So we typically think of this process here, I think this is the way a lot of turf managers learn it, that organic matter inputs come in through either exudates or turnover root material or thatch. It's intercepted by microbes that break it down and then somehow magically becomes soil organic matter. But we know that there's a whole bunch of other trophic interactions involved in these processes, especially including larger invertebrates. These things can, have this traditional view where we know that they're fragmenters of litter. And, I think a lot of this came from the stream ecology literature, where we think of invertebrates as shredders and movers of organic matter. Also in soil specifically being engineers of soil, bioengineers. But there's also this sort of growing recognition of their importance as governors of microbial processes. So we think a lot about this in our lab, like maybe they're just tweaking the way microbial processes flow in soils. We know that a lot of them are fungal and bacterial grazers. They can disperse microbes either on their cuticle or in the gut. So a lot of different potentially really interesting interactions there related to their role in carbon cycling. But in these particular systems, and these aren't the only systems with this trophic level, but especially in turf grasses, we have a lot of root feeding insects, and they can really change detrital inputs to soils, 50 to 30% declines in below ground primary productivity due to root feeding insects. And that's taking a live root zone and turning it into detritus in the matter of a few days. So it's a really interesting component to this food web that we've been trying to understand, not just from a pest management perspective, but also from a organic matter cycling perspective. And I'll get into that a little bit more as the talk goes on. So the first question I wanted to kind of get into was this one of the importance of soil animals and turf grasses. So this is like where I really nerd out. I really think soil invertebrates are the coolest things ever. So we really want to understand better what their role is in these systems, whether it's through microbial interactions, litter fragmentation, soil engineering, any of those kind of pathways. So this has been studied by a lot of people in a lot of different ecosystems, and we've been really interested in just saying, well, do they have the same role in turf grasses? Is it different? These are very different systems in terms of the organic matter inputs. So this is a question that my recent grad student, Natalie Bray, was really interested in, specifically how macro detritivores impact microbial communities and soil processes and lawns. So I thought I would review Natalie's work because I think it kind of gets at this first component really well. So Natalie used this approach of using these exclusion-based mesocosms, and you can see one of the mesocosms here. It's this sort of cage cylinder. So this was a challenge for us in being able to put these things into the soil and keep a live turfgrass system and active roots above it. But we found a way to do that by taking soil cores out, defaunating the soil through a heating and freezing process, and then re-wetting it to field capacity and putting it back into the live root zone under an actively growing turfgrass plug. So roots were able to grow back into these and basically keep it a live plant input-based system. Natalie wanted to contrast these different mesh sizes to be able to exclude or include macroinvertebrates. So we used mesh size, kind of like the Mark Bradford approach to doing that with his litter bag work. If anybody's read those papers, basically just using size-based exclusion to make this happen. So Natalie used a 5 millimeter and a 1 millimeter mesh with defaunated and re-wetted soil within them, buried them below the active turf layer, and then let them incubate for a couple of years with a harvest in each year. So this is a pretty common approach. We kind of adapted it just for this live turf system. And you can see one of the original cores that came out here before soil was defaunated. So this is a pretty typical type of core that gets taken out. This is what you would use to put in a new Gulf hole on a green, for example. We just stole it for our purposes. So this is kind of how it looked. We had harvests of these mesocosms in year 1, and then 2 years after installation, 8 replicates each time. And you can see how they look coming out. Roots from both treatments were growing well into the mesocosms, and vertebrates were really active in them, recolonizing those defaunated soils. And you can even see here, European earthworm kind of boiling out of one of those macro cages. So the first thing we did was we harvested some of these to actually confirm that our fauna treatments were what we aimed for them to be. And so the macro mesh treatments were loaded with mesofauna, as we would expect, mites, columblins, immature insects, and other mesofauna, but also we're loaded with some macroinvertebrates, some millipedes, centipedes, some herbivorous beetle larvae like wireworms, that's the larva of click beetles, and then also scarab larvae, which is probably our most important root feeding insect in turfgrass, as well as European earthworms, so lumbricus earthworms. When we contrasted that with the micro mesh systems, the 1 millimeter mesh systems, and I just highlight this to point out that earthworms were the dominant macroinvertebrate in these soils. So the micro mesh system had pretty much the same communities of mesofauna, really no difference in them numerically or in terms of composition. But fortunately, the mesh was excluding other macroinvertebrates. So we didn't have the herbivorous insects, we didn't have earthworms or anything like that in these soils. So our treatment to actually suppress and minimize those macroinvertebrates worked. And that was the case in both years of the study. So the other thing that Natalie did in collaboration with Jenny Gao-Niffin's lab at Cornell and her student Grant Thompson was to pulse label these plots. So they wanted to be able to, in a separate paper, track the fate of these inputs from the live plants through both years of the study into different soil fractions. So just to see if the presence absence of fauna changes the sort of pathways that these signals follow. So they pulse labeled that with a N15 fertilizer application, and then used the tent-based system to cycle enrich CO2 through to enrich the turf grass. And because these are live plants, over the course of the study, after that initial enrichment, they can dilute. So that's an added challenge that anybody on this call knows if you've done these type of field pulse labelings. Natalie measured about as much as you can think of as a soil ecologist to measure after we harvested these things out of the ground. She looked at microbial community composition, fairly coarse view of it, looking at bacterial and fungal composition. We also looked at microbial biomass using phospholipid fatty acid markers. Microbial activity using a couple different metrics. She did a short-term incubation with soils collected from the field to look at respiration, and also extracellular enzyme activity with the typical fluorescence and absorbance-based plate reader assays that we have in our lab. And then lastly, she also did some physical and chemical fractionation of the soils, basically doing the wet sieving method to look at aggregates and particular organic matter, and then looked at isotope enrichment in those fractions through submitting to the Cornell Isotope Lab on main campus. So just kind of diving into results, the first thing is just an NMS ordination showing differences in bacterial community composition. So the symbols like squares are year 1 and circles are year 2, and then the shading represents macro versus meso. So if they're black, those symbols are soils that allowed macroinvertebrates to colonize. So we saw some differentiation in both years of the study. The differentiation in year 1 had really nothing to do with our treatments, but once we got into year 2, you can see that separation pretty clearly related to presence or absence of macroinvertebrates. So Natalie was trying to figure out, how we could pick out which microbial taxa might be driving this through some vector analysis in the NMS, but also just looking at different taxa. And she found relative shifts in the relative abundance of these 3 groups. So the Firmicutes and Planktomycetes were some of the more interesting ones we saw differences in. But yeah, so some of the individual taxa that responded, not always the same direction to presence absence. In terms of mechanism driving this, we're still kind of juggling different ideas of what might be driving that, but some interesting community composition shifts nonetheless. The story was pretty similar with fungi. It was less of a distinction between years 1 and 2, but you can see in both years we had a separation of the points based on composition. So again, based on whether or not they had macroinvertebrates colonizing the soils. And the taxa that popped out when we looked at individuals was the zygomycota. So we saw in macroinvertebrate inhabited soils, there was a lower relative abundance of the zygomycota. So I mentioned that Natalie also looked at, used PLFA to look at sort of coarse biomass groupings of different functional groups of microbes. So here we looked at lipid markers for saprotrophic fungi and AMF, arbuscular mycorrhizal fungi. And she saw that it was different years that it popped out, but in both cases there was higher biomass of these lipid markers for saprotrophic fungi and AMF in the presence of macroinvertebrates. So despite being fungal grazers, we were seeing sort of I don't know if you want to call it stimulation, but higher presence of those biomass groups in the presence of macroinvertebrates. Same was true for enzyme activities. So here we're looking at phenyl oxidase activity in beta-glucosidase. So phenyl oxidase is a proxy we use for sort of a lignin cycling or recalcitrant organic matter cycling enzyme activity, whereas beta-glucosidase is targeting much more labeled carbon sources like glucose. So in both cases, again, like with the biomass graph I just showed, we saw that the presence of macroinvertebrates led to higher enzyme activities than when they were absent. So not just biomass, but also activity. Natalie found some interesting results with aggregates. So this graph is kind of showing 2 different things, not only our proxy for amount of aggregates, which was macro aggregate mass, but also enrichment, isotopic enrichment. So she found in the presence of macroinvertebrates, like we've seen with a lot of earthworm studies, but also here in turfgrasses, you end up with higher amounts of macroaggregates in the soil. And she found that in years 1 and 2, but also the amount of relatively new carbon coming from that active turf, pulse labeled turfgrass was higher in the presence of macroaggregates. So more new carbon coming into the system, ending up in those macroaggregates. So not just a total aggregate response. So we thought that was quite interesting. So like we've seen in other ecosystems, especially from the forest invertebrate literature, these invertebrates in our turfgrass soils are altering the composition of the community and its diversity, stimulating the size and activity of that community, And interestingly enough to me, promoting the transfer of that new organic matter coming in from photosynthate into aggregates and other soil fractions. So a really interesting role for these invertebrates. And this is kind of unique because in turf grasses, even European earthworms are seen as a pest. So they really struggle to manage them, especially on putting greens. So this is interesting data and hard to present to the turf industry because of all the benefits that they also kind of bring to the system. So trying to walk that line between how you manage them as a pest on something like a putting green, but also try to promote their benefits out in the fairways and other parts of the soil, like in lawns, is a unique stakeholder education challenge. So looking back at this here, it's pretty clear that these invertebrates are playing interesting roles in these systems, especially in association with the microbes that are there. But again, we're really interested in how is the management that we're carrying out in these systems affecting those communities and the processes that they drive. So I want to talk about some of that work next, because this is kind of where I've become really active over the last 5 years or so in my program. So there's a lot of variability in pest management practices and intensity and what drives decision-making for pest management. So it's an area that I really wanted to get into and try to understand better in terms of how soil health, soil health and soil food webs are responding. And we've done this and everywhere from lawn systems to high-end golf courses that have a lot of synthetic inputs to them. So we've come up with kind of a suite of soil functional proxies that we use to try and capture the responses of these different practices. So obviously, because we focus so much on invertebrates, using Berlese funnel extractions to characterize arthropod communities, we look at enzyme activities as well as a proxy. For a while, we've been looking at mycorrhizal fungal root colonization as well. So AMF colonization in turf roots, that's something that stakeholders understand, and it's really important in their mind for stress tolerance. So it's something that we've been quantifying as well as an ecosystem service too. And then also entomopathogenic fungi, which are important biocontrol service providers for pest suppression in these systems. So we have an assay to assess sort of the infection potential of the soil in providing that pest regulation service. So the one project I want to talk about now is one that we did on golf courses that were across New York State. We kind of ranked golf courses based on pesticide input or management input intensity. And we ranked them in a bunch of different ways based on the frequency of inputs, the cumulative inputs, as well as the identity of the active ingredients that these golf courses are using. And we used an index called the EIQ, or environmental impact quotient, in that sense. The takeaway was that no matter how we arrange them, frequency, cumulative amount, or the EIQ, they binned this way. So we ended up with 3 separate bins. Some golf courses that have very little money in their budget, so they don't do any pest management. Others that have low input, low frequency, low concentrations, and also these high input golf courses. So those 3 separate bins is how we broke it down. And all the sampling we did was in golf course fairways, so nothing on intensively managed greens. So this is just sort of some of the interesting takeaways from that. We found some interesting responses in most of the metrics that we looked at, but here's some that I thought would be worth mentioning. The first is the response of acid phosphatase. We saw that, and this was a study we carried across 2 years, 2 times a year. We found that in general, there was a suppression of acid phosphatase activity. Acid phosphatase is sometimes used as a proxy for overall microbial activity as well, although we think of it more as a related to phosphorus cycling. So, but in general, as intensity increased, we saw suppression in these activities. You can see the significant response was here in spring of 2014, but the same pattern kind of stuck around all seasons in both years. We saw a similar response in mycorrhizal fungal colonization here, and you can see a couple of significant dates popping out. So in some cases, it was only at the really high intensity level, like fall of 2015, way over here. But in others, no matter what the rates or frequency inputs, if you were managing with pesticides, we saw suppression in mycorrhizal colonization. And this one has kind of struck a chord with the industry because they are so interested in mycorrhizal colonization of their turf grasses. So there are trade-offs that come with the intensity with which they're managing these courses. So the other area that we've kind of tapped into was a bit more nuanced view of pest management. So rather than just overall pest management intensity, we wanted to really look at some really common practices and intensive practices that are used to manage turf. So the one we focused on was annual white grub management. So annual white grubs are things like Japanese beetle, European chafer. In this area, probably oriental beetle is one of the more common ones. So they're really aggressive herbivores and feed pretty actively in a couple different times of the season. Right now, soil temperatures are increasing and they're about ready to come back up into the root zone to feed for about 2 to 4 week period before they pupate. And then if you look all the way at the left of this illustration, those insects pupate, emerge above ground as adults where they'll reproduce. Some of them will be pests and feed as adults, but not all species. So then they'll reproduce, the females will burrow into soil to lay eggs, those hatch, and then there's this window from August through October that they'll feed again. And this is kind of the window of management. So this is a pest that crosses all types of turf grass. It's a pest in home lawns, it's a pest on golf courses, sod farms, cemeteries, athletic fields, schools, you name it. And often for a number of reasons, but partly because this is a very affordable approach, people will manage them in this very preventative way with systemic neonicotinoids. And I'm sure everybody's familiar with hearing about neonics and the controversy around honeybees and pollinator decline in general. But the neonics, it's the most affordable. It's very cheap for someone to make wall-to-wall applications of these products. Doing it preventatively doesn't give you all the opportunities to use proper integrated pest management, and we'll talk about that in a bit. But we wanted to really look at this practice and see, well, what are the trade-offs when we're using this really common pest management approach in these systems? So just to kind of describe the approach a bit more, it's preventative in that this window here where the adults are present, that's when the products have to be applied. And the reason for that is because it's a systemic product. It has to be placed in the root zone, so it will interact with roots. And so by the time these eggs hatch, it's in the roots and it's expressed throughout the plant. So you need it to be there and taken up by the time the eggs hatch. So it can be really effective, but you have to put it out before you have any information about the larvae that you're gonna get. Are they there? Where are they? And what species and in what density. So you don't have any of that information when you're making these applications. So it really kind of takes it out of alignment with a balanced integrated pest management approach. And we found that I think it's like only 25% of the times this product is applied, it's actually necessary to have applied. So it's over applied a lot, and that's partly because of cost. But this is a slide that takes some background to explain, but this was a 2-year study where we wanted to see after these neonicotinoids, specifically imidacloprid, which is a really common neonicotinoid application, how long do they stick around? The idea is that they go into the soil, they're taken up by plants, and then they're in the plant. But we know that's not a perfect process. And so we wanted to see how much of it stays in the soil. So here I'm just showing the second year of this study. But this -4 on the x-axis, this is actually before we had made an application in the second year. So this shows that we had carryover of imidacloprid in soil from the previous year, and actually pretty strong carryover. So nobody's even aware that this stuff is still sitting in the soil in the spring before you make another application of that active ingredient. So we made a spring application, again, timed with when those adults are present, and you can see the active ingredient concentration spike again. We were looking at 3 different label rates of this product. And then you can see it slowly declines in concentration, but even once we're out 16 weeks after that soil application, the material's still there in detectable amounts. So it sticks around quite a bit. How well it's taken up by plants or broken down by microbes varies a lot by soil conditions. So it's not just a quick in and up, taken it by the plant and out of the system kind of process. So it's one of the reasons we wanted to see what its impacts were on biological processes. So the first process we looked at was, or the first biota that we looked at were mycorrhizae again, and this time we're looking at root infection rates from arbuscular mycorrhizal fungi. And here we're looking through, again, the same time series, and we found that at 1 and 8 weeks after the application of those products drenched into the soil, you have a suppression of mycorrhizal colonization of the turf roots. So it's not every week, it's kind of sporadic when it shows up, but some significant windows of time where you may have compromised mycorrhizal communities colonizing the roots. We found some similar responses when we looked at the invertebrates. So here's some data on Columbella. We didn't see any responses with mites or the acari, but or other arachnids. But when we looked at insects or their relatives, like columbullans, we found some significant suppression of their abundances. So here it's again the same 3 rates we looked at of the imidacloprid application compared to an untreated control that just received water. And at 1, 4, 8, and 12 weeks following the applications, we had suppression of the abundances of those communities. So once we got out to 12 weeks, there was some recovery, especially in this lowest rate here, but with the mid and high rate, and honestly, the high rate is what everybody uses, it was still significantly lower abundances in those lawns. So it's something that's having sort of lasting effects on these communities. And you can see some rebounding, but a pretty significant amount of time where these biota are at lower densities in the soil. So related to this, I've got a student who's been really interested in Columbella, and the turfgrass textbooks call them thatch busters and says, oh, they're really important in this process, but nobody's really measured that well. So we wanted to see if they in fact do that. So Hayden worked with the same columbullans. Hayden's just starting his 2nd year with me now. And he was working with the same columbullans in the lab to see how much they actually contribute to thatch decomposition. And it's pretty detectable. So when those columbullans are present in turfgrass soils, they increase thatch decay significantly. So they are playing that role in there. So those pesticide-driven changes could even have implications on something like thatch, which is a huge management concern for turfgrass managers. And sometimes they even have to manage it physically by raking it out of the soil. So if any of you have ever used one of those thatch rakes, you know what I'm talking about. So coming back again to this framework, which I seem to like a lot, there's some clear some clear consequences that come from the pest management practices we use, whether you're looking at overall intensity of the management or individual sort of specific management practices targeting things like white grubs can have some negative impacts. We have some data showing they can compromise insect pathogens in soils as well, especially with more intensive pesticide inputs. So across this sort of beneficial biota and ecosystem service realm, there are very real trade-offs that come with the decision-making that we are throwing out there. So some people ask, well, we just stop managing white grubs? Why do we have to do this in the first place? And I mean, there are some systems where they're never managed, like my lawn. And I have lots of white grubs in my lawn, and sometimes it looks kind of cruddy, but there's many times where they're there and you really don't see a whole lot of damage. So there's a lot of push to kind of move in this direction and stop managing them altogether. But because of their impact on carbon inputs to soil, we were really interested in that question and just finding out, well, what role do these insects even play? Is there a carbon side to this story, or is it really just about aesthetics and getting rid of pests? So my recent postdoc, Hui Gan, and another sort of short-term postdoc in the lab, Chao Liang, tackled this question as well, looking at what role these insect infestations and root-feeding insect activity play in shaping microbial activity and carbon cycling in these lawns. So this kind of came from some observations we made of feeding rates. So we looked at my hand up here. This is a picture of Japanese beetle frass or feces you can see scattered throughout this sand. And it can be really concentrated in areas that we have sort of high infestation densities. So their production rates are also really high. So over an 8-day period, you're getting 60 or so fecal pellets built up per larva. And when you look at the outbreak densities that you can have for these insects, it can be quite a bit of material. It's like 150 milligrams per meter square of organic matter that's going from live root system to detritus in this sort of frass pool instantly. So it seems pretty significant. We did some preliminary incubations to look at CO2 responses to spiking soil with these different inputs. We compared the addition of just the equivalent amount of roots to an equivalent amount of frass or feces that you would get over an infested area. And we found, as you would expect, an increase from controls that are receiving no inputs, but the frass led to a greater CO2 accumulation than the roots in general. So there's something unique about it compared to just the roots that are not passing through the gut that drives even greater stimulation of microbial activity. So Huitia and Chao wanted to tackle this a bit more and conduct a field study. We mimicked what you would do in kind of a pesticide trial where you cage insects in these PVC arenas. So they drove these PVC arenas into the ground and established different densities of probably the most common white grub species that we have in turfgrass in New York State. Papillia japonica, or the Japanese beetle. And these were third instar. That's the most damaging stage of the larva. That tends to be late summer to fall that they're present. So we collected them and created these different arenas with different densities of those larvae. So either no larvae or 6 larvae per arena were the extremes in our treatment. They went out in October. As I said, that's kind of like the window of time where they're actively feeding before winter kicks in, and they go below that frost line to hide from the winter temperatures. We let them overwinter, and then in May, they come back up and feed again. So we let them feed for that window before they pupated, and then harvested everything out. So 2 windows of time where those larvae could feed. And then, like Natalie's work, Huetia measured everything under the sun she could think of and possibly do in our lab. Soil carbon and nitrogen content through just dry combustion analysis. She looked at microbial biomass, this time with just chloroform fumigation, same enzyme assays that Natalie did, and also looked at microarthropods, because we cannot seem to not look at microarthropods in our lab. So pretty much across the board. And here's what we found. So the first graphs are just showing you the biomass response. So Huetia found that soils with P. japonica present at those densities had significantly higher microbial biomass. And we saw that reflected in both the carbon and nitrogen fractions, which was interesting. Looking at enzymes, the enzymatic response sort of varied by the substrate we were looking at. So phenol oxidase activity was suppressed or was lower in the presence of Japanese beetle, but cellobiohydrolase or the cellulase activities were higher. So, we think that might be because the frass is so much more labile than the other organic matter there, that maybe the need to break down recalcitrant substrates was a little bit lower. So hence the increase in hydrolases and decrease in oxidases. That's our hypothesis anyway. So looking at the invertebrate community, we found one really interesting increase, which was with the orobatid mites. And this is not any particular orobatid. We had probably about 4 or 5 different taxa in this category, but we were even seeing this reflect kind of at higher trophic levels where the microarthropod numbers were increasing in the presence of this root feeding insect, which was very interesting given the short time frame over the study. And then lastly, we looked at total carbon and nitrogen stocks in the soil. And this again was just dry combustion of bulk soil from the outside the root zone. And we found in both cases that those levels declined in the presence of Japanese beetle. So stimulating all this quick microbial activity, which looks like it might even be leading to a decrease in the overall stock of carbon and nitrogen in the soil itself, which was a kind of a surprising response. So this relationship between root feeding insects and carbon is not brand new, but I think the directionality might be kind of new. We've always tried to model these factors that might predict hotspots of pest activity. So for a while, we thought carbon might be a good predictor of that. And it's, where you have high carbon, you tend to have low densities of root feeding insects, particularly white grubs. And there are a number of studies that have looked at this, but there's still a lot of variability around it. It's not a perfect relationship that helps managers predict where pests are. But our data, I think, muddy those waters a bit more, because it kind of shows that they might actually be driving this chemical landscape more than, in Mark Hunter's words, the phytochemical landscape, more so than responding to it. So it might be both at play, but we have, I think, some neat evidence that they might be shaping it or influencing it to some extent on their own. So I won't go through every bit of this, but Huetia came up with this really interesting framework to sort of fit root herbivores into this process to show that, how they influence the detrital community as well as the soil organic matter fate and sort of process. So, they're not a perfect fit into any food web, but I think it's really interesting to think about it this way when they're shifting live to detritus very quickly through herbivory. So I'd love to chat about that more with anybody who's interested. So I don't know where we're at on time here, getting a little close, but so in my work, we think a lot in my lab. I have people in my lab that are working on really basic soil biology, all the way to very applied work, trying to optimize pest management. But we think a lot about this framework of trying to find this balance between meeting the needs of pest managers as our stakeholders, but doing so in a way where we're, I'll say, exploiting or optimizing and capturing the benefits of soil health and ecological services. So this is something that really interests me, and trying to find ways to meet these goals that we have, these very practical bottom line type goals that this industry has, while helping them harness and embrace soil biology. So there's a number of different approaches that we're taking to doing that. And I thought maybe it would be fun to just kind of go over how we're tackling that a bit in my extension role. because some places it's an easy sell and others I think it's going to be a lot longer term relationship before we really make good change there. So the typical ways that my lab is approaching this is in some cases just through basic IPM training. And I'll talk about what I mean when in a bit regarding that. But so this is the integrated pest management framework, which I think shares a lot of similarities with soil health management and soil health promotion. So I'll talk about that. I do a lot of educating of turf grass professionals on soil biology, basic soil biology. And sometimes I have to like sneak it into my talks on pest management, but there's a big push in my extension program to educate them more on the broader soil biological community. A really big part of this is identifying stakeholder barriers and opportunities. This is really important for making sure you're asking the right research questions and framing your programming accurately. So I'll talk about some of our efforts to do that. And then also educating future stakeholders. So this is kind of where it crosses into the outreach realm, but in the system I work in Turfgrasses, I think that outreach and extension, it just depends on how old the person is, that they're going to be future homeowners or future renters, future turfgrass users. So I think there's a really clear linkage there where we're educating them as stakeholders and future stakeholders. So I'll talk about that as well. So this first one, integrated pest management, we've had a lot of different activity here. Some of this is just me giving lots of extension talks about how to manage pests properly. And this, you can create a lot of sustainability gains even in just doing this, because if you look at neonicotinoids, for example, I mentioned that they're often over applied. Teaching people the proper timing and proper decision-making for making those applications, you can make a lot of sustainability gains just through doing that. So a lot of my extension talks are focused on how to actually use these products right. Sometimes it's to homeowners and master gardeners. Other times it's to a group like this at Doubleday Field of turfgrass professionals. So that are managing athletic fields, managing golf courses, managing school facilities, cemeteries. We do hands-on training with this massive insect teaching collection I put together to teach people how to do proper diagnostics and decision-making around pests. So we quiz the heck out of them with these teaching collections, and they get pretty good at it. We make them use a hand lens to identify these things and hope that they're carrying some of these practices over to their careers, although that's been harder to capture, that sort of adoption change. But this is a big way that I spend my winter, traveling around with this teaching collection to train folks on that. To aid in that, we've built some online keys. So we built this interactive key to identifying white grubs, because a lot of people that have been managing grubs for decades still don't know how to do this. And there's a lot of decision-making you can do to determine situations where you do and absolutely don't need to manage white grubs. So part of that is their identification. So we built this key, it's sort of a adaptable platform so they can use it on a smartphone or desktop or tablet in the field to identify them. Our other efforts in this realm have been trying to make some of those barriers a little bit lower. So, even if we teach them, give them a key to manage grubs and identify them and put them through these workshops, it's still hard to convince them to bring that into their timeline and put it on their calendar and do it. So we've been trying to make some of these things just lower the barriers for it through some of our pest management work. So my recent grad student, or my newish graduate student, Morgan, has been trying to find ways to just improve the ability to introduce biological control organisms. And she's been working with biocontrol fungi. So rather than forcing the practitioner to learn how to handle and use these fungi, we've been trying to introduce them as a seed coat to see if we can get them to establish coming in when they seed their turf. So we kind of remove that piece of their management for them. The other way we've been doing this is trying to make the scouting and decision-making a bit more automated. So I have a postdoc, Louise Roberts, who is only going to be with me through August now. She just got a job back in the UK. But she works in bioacoustics and biotremology, and she's been doing work with these technologies in soils for us with annual white grubs and other soil animals to see, can we actually use acoustic sensors to detect and characterize hotspots of pests. And as I was talking about before with identifying what drives these hotspots, we still struggle with characterizing that for these pests. So Louise has been doing some work to see if we can distinguish different invertebrates through their acoustic signatures and actually characterize communities, but also pick up on where hotspots occur in these managed soils. So most recently, Louise has been working down at Greenwood Cemetery in Brooklyn, And this involves like working from the really anthropogenic loud edge of the cemetery into the quiet interior to see if we can pick up the signal from these organisms, even in really noisy conditions where there's a lot of anthropogenic sources of noise. So that's been a fun project, totally outside of my wheelhouse. So if you have any acoustic questions, I'm going to sound like a moron when you ask them to me. A very interesting area, and one that could really help move our scouting practices forward with the industry, I think. I mentioned also just general soil biology education for turfgrass managers. This is a fun one for me, but as I said, I have to try and integrate this into some of my more standard talks, but I do a lot of this where I'll just sneak some basic education about soil biodiversity and the ecological roles that these organisms play in turfgrass soils. And I've found that, you know, there can be really high biodiversity in even some of these heavily managed turf soils. So it's something that we can really kind of champion with them as something that they can hold up and showcase if they're interested in it. But so this is something sort of like a long-term education goal I have with the turf industry. And the next that I mentioned was trying to characterize these barriers and opportunities with stakeholders. So our biggest push recently in this realm was to better understand the motivations of homeowners and how they manage their lawns. So I have a collaborative group in New York, Ohio, and Florida working on this with me. And we developed a homeowner survey to try and capture some of that information about them. What motivates them? How interested are they in trying to manage their lawns for environmental benefits versus aesthetics and other characteristics? We got just over 300 participants for the survey that we put together by state. So we had 900 some participants in the end. And again, yeah, getting at beliefs, practices, preferences, and expectations for their loan. Just to kind of wrap up some of the other stuff we've been doing, educating future stakeholders. This is a really big interest of mine, doing a lot of outreach. We have a line of support at Cornell called EOA, or Extension Outreach Assistantships. And I was able to support one of my recent grads, Max Helmberger, on this. And he just went nuts, creating a whole series of clay animations on soil food webs. And if you're at all interested in this sort of stuff, let me know. It's very easy to find if you Google Max's name in YouTube. Everything that you can think of in the soil, from like general soil ecosystem services through life cycle of the squash bee, he has a clay animation on. So really cool resource to check out. And this is something we push with school kids a lot to kind of watch these videos and use them in their school activities. We've created an entire lab on how to use school athletic fields for measuring soil ecosystem services, particularly biological pest regulation. So we have a lab available for middle school science teachers. And we aligned it sort of with their New York State learning goals. So it's hopefully a little easier to slip into the curriculum rather than make a major change. So this is also available on our website. And Max, as part of his EOA, also created this really nice soil animal handbook. So it's kind of a guide to the good and bad critters in soil, all the way to how to collect them and keep them as pets in your house. So a lot of really cool resources in that book. And we've done this in a number of different settings, kind of like come get the resources if you want it style, all the way to us doing outreach work with local schools. So we've done this with schools across New York State. This school athletic fields lab in particular, we led with 3 different schools, and they just got really into it. So doing assays in their lab to look at infected larvae that kind of rot and get all nasty in their teachers' benches. But all of this activity as well, the lab and the guide for the lab is available on our website for anybody interested in outreach. And I know a lot of folks here at Cary are interested in outreach. So yeah, that's kind of our effort to take who's going to become our next stakeholders down the road and show them a different side of these turfgrass systems for the biodiversity they can host. So with that, I know we're pretty much right at the end here. So I just want to thank all of you for having me here. And for those of you who aren't necessarily at Cary, but logged on to see this, and whether you've got questions now or later, I'd love to entertain them and chat with anybody. So, and also just want to thank my lab, because I didn't produce any of this. My lab did it all. So, so yeah, thanks very much. I appreciate your time.
Thank you so much, Kyle. It's great to see what you all are up to. And I particularly am excited to see what's happening here in New York. So I know we are tight on time, but I will ask one question in general, which is, for a lot of your management decision-making, do you think that a big way of fixing it will be on these higher order, like legislative decision-makings, where we kind of take it away from the individual? Or do you think that you can make quite a bit of progress working on a more pointed, like individual changes on a lawn scale.
Yeah, I think that's a good question. So I think some of it's probably going to come from the top down. Like I'm pretty sure that's what's going to happen with the neonicotinoids. So they are from New York being removed this year from the homeowner market. So none of the products like grub killer products that are out there that you would find at a big box store are going to contain neonics anymore. But they're still available for the professional market. So you could have a lawn care company that could come in and still apply them. So that is definitely happening. And the other active ingredients, it's not just the neonics, there are others that are being removed in sort of that top-down fashion. But I think there's other situations. There's a lot of interest from homeowners that they don't know exactly what they want. They just know they want something that's a bit more diverse and ecosystem service related. So I think that if we could link up what homeowners are interested in with the risk management model of some of the landscape companies and lawn care companies, so they help manage their risk on that end, I think it could happen from the inside a little bit more there. And that's actually an area, I mean, that's where most of our acreage of turfgrass is. It's in home lawns. So I think if we could make that connection better, that would be a place to do it. So it's not so top down, because the industry obviously doesn't receive it as well when it's coming from top down. It's a lot harder pill to swallow. So yeah, I think it's going to be a mix in the end, Jane.
Yeah, that's great. Well, I know that people are going to need to jump off here, so I'm going to let you also say goodbye, but thank you again. Okay. And I know that Kyle will be around on campus. So for those of us at Cary, if you have your questions, you can always interact with him at lunch or later in the day. But thank you so much for the great talk. And thanks everyone for tuning in and sticking with us in our up and down internet. So, all righty. Have a good day.
Thanks, everybody.